Welcome back to this Biosystems Analysis and Modeling series. In the last module, we looked at some systems and explored how to represent these systems diagrammatically and mathematically. In this module, we will examine some common representations for specific phenomena often observed in biological and ecological systems. Upon completing this module, we will be able to, first, understand saturation processes and representations in models. Second, understand the law of mass action and its representation in models. And finally, you will be familiar with spatial processes of advection and diffusion and their representations in models. The first generalized process that we will consider is saturation. Saturation is a commonly occurring rate-limiting phenomena that we can easily represent in several mathematical forms. It applies when the rate that a process can proceed is limited by availability of what we will generally term reaction sites, which can be interpreted broadly as opportunities for interactions between the components required for a process to proceed. Saturation kinetics, the equations describing how fast a saturation-limited process actually proceeds, were originally developed for enzymatic systems, but applied to a broad range of phenomena. We will start by looking at enzyme systems to develop the conceptual model. Enzymes are typically proteins that facilitate a reaction by combining with a substrate reactant to form an enzyme-substrate complex, which in turn generates the products of the reaction. Hence, the reaction requires the availability of enzymes to serve as active sites, allowing the reaction to occur. As a result, the rate at which a quantity of enzymes can process a quantity of substrate is limited by the availability of these sites. When the quantity of enzymes, or in other words, the number of possible sites, is large compared to the quantity of substrate being processed, substrate has little difficulty in finding a reaction site, and the rate of the reaction is therefore high. As the reaction sites become clogged, any substrate looking for a reaction site will have more and more difficulty finding an unoccupied site, and may need to wait until a site becomes available, thus slowing down the rate of reaction. At low concentrations, the reactants are generally there are plenty of sites available, and the processing rate is relatively unaffected by the limitation related to the availability of reaction sites. As the concentration of reactant increases, the rate of reaction increases because there is more reactant available to process. However, as we continue to increase the concentration of the reactant, the rate of the reaction will continue to increase, but at a decreasing rate, until finally the system becomes completely saturated. Adding more reactant will have no effect on the processing rate, since the system has exhausted its processing capacity. To capture saturation phenomena in models, we need an appropriate mathematical formulation. One such formulation is the Michaelis-Menten model, widely applied in enzymes kinetics, as well as other systems governed by saturation. We will look at that next. Michaelis-Menten kinetics represent the enzyme saturation phenomena as a simple hyperbolic function, shown here. The basic behavior of this function is that at low concentrations of the substrate driving the reaction, the velocity of the reaction, or how fast it proceeds, is relatively small because not much substrate is available. The rate-limiting factor is the lack of substrate to react. As the substrate concentration increases, the rate of the process increases as well, but at a decreasing rate as the rate-limiting factor begins to shift towards the availability of reaction sites. Finally, at some point, increasing the substrate concentration has no effect on the reaction rate. The system has become fully saturated and additional reaction sites are unavailable. The Michaelis-Menten kinetic model has two parameters, Vmax and Km. Vmax is the maximum rate of reaction of the system, occurring when the system is fully saturated. Km is generally referred to as the half-velocity coefficient and represents the substrate concentration where the reaction rate proceeds at 1/2 of the maximum rate. The Km parameter determines how sensitive the system is to substrate levels. The Michaelis-Menten formulation has been used in a number of applications outside of enzyme kinetics because it captures the concepts of saturation well and involves only two model parameters to fit. Another commonly used saturation function is the Monod equation, a very similar formulation to the Michaelis-Menten equation. It is used to describe the effect of rate-limiting nutrients on the growth rate of a population. For example, in a bioreactor using bacteria to convert nutrients to a desirable product, it would be typical to model the growth process using a hyperbolic Monod equation as shown. Note the similarity with the Michaelis-Menten equation. Mu max, the maximum growth rate of the population, is the equivalent of Vmax, the maximum reaction rate in the Michaelis-Menten formulation. And Ks, termed the Monod constant, is the equivalent of the half velocity coefficient, Km, in the Michaelis-Menten formulation. Another example of a hyperbolic function capturing a saturation process relates to foraging success. Foraging success has been modeled with a variation of the Michaelis-Menten equation called Holling's disk equation. You can see from the curve shown on the chart that the general characteristics of this function capture the saturation concept. In this case, the y-axis shows the rate of successful foraging, RF, as a function of the concentration of the prey being foraged upon, shown on the x-axis and labeled x in the rate equation. The idea is that foraging success increases with prey density, but as prey density increases, the forager's ability to actually process the abundant prey becomes the rate-limiting factor. Oftentimes, a saturation effect is just one part of the rate at which a saturation process proceeds. If there are multiple factors that interact, it is generally appropriate to combine the effects multiplicatively. An example is Steele's equation for photoinhibition of photosynthesis. This equation captures two factors influencing the rate of photosynthetic production in a photosynthesis-driven system. For example, a pond for producing algae. In this system, light drives the photosynthesis rate, but this rate can become limited as the algae's internal machinery for processing additional increments of light becomes saturated. At lower light intensities, with the absence of photoinhibition, the model says that photosynthesis rate increases linearly with light intensity. However, as light intensity increases relative to an optimal intensity level, the algae's capacity to process more photons becomes saturated, and in this case, very high intensities can actually result in a decreased photosynthesis rates as the photosynthetic machinery begins to shut down to avoid damage. A common scenario is the uptake of multiple nutrients or substrates, and we want to model the combined effects of these nutrients on the overall growth of a population. For example, consider growing a crop where the crop requires both nitrogen and phosphorus to grow. One might be limiting, or the other, or perhaps both might be limiting. We can apply the concept of saturation to modeling the combined effects of these two nutrients. Several alternative formulations come to mind. The first you may recall from ecology, Liebig's law of the minimum. This law states that if multiple nutrients are required for growth, the one that is most limiting will control the growth rate. The others will have no effect. We can capture this concept using saturation kinetics as shown here by calculating the individual saturation influence effect, determining which one is most limiting, meaning a lower S over Ks plus S ratio, and ignoring the others. Alternatively, we might have a situation where the nutrient limitations interact with each other. In this case, an appropriate model might be a multiplicative relationship between the limiting factors, indicating the combined effect of individual nutrient limitations both influence the overall rate of growth, unlike the Liebig model we looked at previously. A third representation might average the two individual limiting effects. The model in effect says that the limitations are additive. They do not interact with each other, but they both contribute to the overall limitation on the growth rate. You can see from the plots that each of these models and their embedded assumptions result in similar general forms, but each has nuances that result in slightly different results. Which one of these is best for representing multiple nutrient limitations? The answer is, it depends. Understanding the different assumptions that these three models make is key to selecting an appropriate representation for a specific system. Another useful systems concept is the law of mass action, originally developed to describe reaction rates of aqueous chemical reactions. This law states that the rate of reaction is proportional to an integer power of the concentrations of all substances taking part in the reaction. If we have a reaction involving two reactants, for example, P and Q, the law of mass action states that we would expect the rate of the reaction to be proportional to the product of the concentrations of P and Q raised to the power of the stoichiometric coefficients associated with each reactant. For example, consider the aqueous reaction of hydrogen ions with carbonate ions in a solution. These react to form CO2 and water. 2 hydrogen ions and 1 carbonate ion is needed for this reaction. According to the law of mass action, the rate at which this reaction will proceed is proportional to the product of the hydrogen ion concentration squared and the carbonate concentration raised to the first power. The hydrogen ion concentration term is squared because the stoichiometric coefficient for hydrogen is 2. A key insight of the law of mass action is that for processes involving an interaction between two or more quantities, the rate of that process is generally a multiplicative function of the quantities required for the reaction to proceed. The law of mass action can be easily incorporated into our state equations describing system dynamics. For example, consider the reversible chemical reaction shown here. We could model the mass balance of, for example, the product C by applying the law of mass action to the forward and reverse reactions. Our state equation includes the additions to C, captured in the forward reaction in the first term of the differential equation, and the reverse reaction, captured in the second term of the differential equation. This concept can be applied broadly outside of chemical reactions. For example, the Lockable-Terra predator-prey model that we examined previously is an example of the application of the law of mass action. The prey growth term is conceptualized as requiring a single unit of population and no other reactants to proceed. The reaction rate, really, the growth rate, is proportional to the concentration of the reactant, a unit of prey. The second term represents loss due to predation, where predation can be thought of as an interaction involving two reactants, a predator and a prey. The law of mass action suggests that the rate of the loss via predation is proportional to the concentration of these two reactants raised to the first power, since only one of each is required. Similarly, the predator mortality term is conceptualized by the law of mass action as a reaction involving only a unit of the predator, with an appropriate proportionality constant, the per capita mortality rate. We will close this section with a discussion of spatial flows and how we incorporate these into models. We will focus on two types of spatial flows, advection and diffusion, and explore how to incorporate dynamics associated with these flows with additional transformation processes. Let's start with advection. Advection is the flow of material from point to point in a linear fashion. Think of a river moving downstream carrying a contaminant or fluid flow along a long, narrow bioreactor. Assuming a flow rate f, we can model the change in concentration of a state variable flowing in the liquid at any point in space with the relationship shown here, which indicates that at a given location along a flow path, the rate of change of the concentration is given by the flow rate times the concentration gradient of the state variable through space, at that location in space. This is an appropriate equation when the flow rate is reasonably constant over the system in question and applies to one-dimensional flow only. The advective flow equation states that the rate of change of the contaminant C at any given point in space is a function of the flow rate and the gradient in space of the contaminant. The effect of this is to drive the contaminant downstream in the direction of the flow. Diffusion is another spatial process that moves mass through space, in this case in response to the differences in concentration gradients. The rate that this process happens is controlled by a diffusivity constant, d, that varies with the materials involved in the diffusion process. While typically used to analyze substrate flow in liquids, the diffusion equation has been applied to a variety of other phenomena, such as the diffusion of knowledge through a population. The net effect of diffusion is to level out any concentration gradients through time. Solving the diffusion equation results in the response shown here. Again, we will start with a pulsive contaminant to start the clock ticking. The diffusion equation states that the change in contaminant concentration through time at any location in space is proportional to the second derivative of the concentration gradient in space at that location. So locations with high negative curvature have relatively fast reductions in concentrations, while those locations with low positive curvatures slowly increase in concentration. The effect of this is a flattening of the concentration curve through space as the diffusion process proceeds. Note that the area under the curve remains constant through time. Ultimately, as the system comes to steady state, where dc dt is equal to 0, the concentration gradient must also be 0, indicating a uniform concentration throughout space. We can easily combine the effects of advection and diffusion. Because these are independent processes with no interaction, they are additive, and our mathematical model reflects that. An appropriate application of the combined advection-diffusion equation might be a slow-moving river system where the rate of substrate movement resulting from diffusion is in the same order of magnitude as that resulting from advection, so both processes would need to be considered. Looking at the response of the combined advection-diffusion equation through space and time, we see the additive effect of the two processes. Advection drives the contaminant uniformly downstream, while diffusion simultaneously flattens the concentration distribution through space. We can easily add material transformations into the mix when we have systems for which advective flow processes, diffusion, and transformation are occurring simultaneously. We simply add a reaction term to the processes influence the dynamics at a point in space. For example, let's consider the reduction of a nutrient in a plant-based treatment system reactor. We can easily add material transformations into the mix when we have systems for which advective flow processes, diffusion, and transformation are occurring simultaneously. We simply add a reaction term to the processes influencing the dynamics at a point in space. For example, let's consider the reduction of a nutrient in a plant-based treatment reactor where the plants are growing in a long channel and we want to examine the nutrient profile through time along the channel length. If the water in this system is relatively slow moving, we would expect both advection and diffusion to be important in this system. Further, we might expect nutrient uptake by the plants to be a saturation-dominated process. Our final model would include an advective term, a diffusive term, and a reaction term capturing the saturation kinetics for the plant uptake of the nutrient, in this case using the Monod equation. The effects are additive because these are independent processes that don't directly interact. Solving this model would provide the expected behavior of the system through space and time. Finally, we note that the prior models of advection and diffusion consider one spatial dimension only. We can easily extend these equations to multiple dimensions by simply adding terms reflecting dynamics in each of the dimensions to be considered. However, we experience increasing difficulty in solving the resulting equations, both in terms of having analytical solutions available and in terms of computational expense when solving these equations numerically. Complex models in time and 3D space can be extremely computationally intensive, sometimes taking hours, days, or weeks of computer time to solve on large domains. So, to recap, in this module we explored a few mathematical approaches for some fundamental processes you are likely to encounter in biological, ecological, and hydrologic models. Specifically, we have looked at saturation, the law of mass action, and advection and diffusion in terms of spatial flows. We saw that saturation is a common phenomenon controlling the rate at which processes can occur. We examined a variety of mathematical representations appropriate for representing saturation processes, all of which were variations on a hyperbolic function, and explored how individual saturation functions can be combined when multiple limiting factors may be present. Second, we explored the law of mass action as a way to conceptualize the mathematics of interactions. Specifically, the law of mass action states that the rate of a process proceeds is proportional to the multiplicative relationship between the participants in the process. When processes are controlled by interacting variables, the rate of the process is governed by the product of the concentrations of the interacting variables. We saw an example from the chemical reaction perspective, but also saw how the same principle can be applied to biological systems like the Lockable-Voltaire predator-prey model. Third, we explored spatial processes of advection and diffusion. Advection moves material through space in response to fluid flow. Diffusion moves material through space in response to concentration gradients. We examined mathematical formulations for each process and identified situations where each process might be important. We also discussed important transformation reactions and how to incorporate these with spatial processes to look at both transport and fate of materials in aquatic systems. One thing we didn't do was discuss how to solve the models that are distributed through space and time. We will cover some basic techniques for solving these multidimensional models in the next module.